So on the last video, we ended on a bit of a downer, and I just want to reassure you it's not all over yet. There are many organisations supported by academic studies that are intent on trying to save what we can and scale back our impact on the planet. You can always research the case studies we've discussed or find your own and have a look at what organisations are trying to do to stop further decline. There are lots of good websites for this, such as the WWF, the Nature Conservancy and the Woodland Trust. Now they're a small selection in a vast number of conservation trusts, so do take a look at those. However, we can now move on to bioinformatics. Bioinformatics is the application of computational tools to analyse biological data, so genetic and protein sequence. So genetic sequence will simply be a long string of A, T, Cs and Gs, and protein sequence will be a long string of amino acids. It's a very new tool to conservation biology. Bioinformatics helps conservation in a number of ways, but two of the main ways are providing freely available databases to store genetic information collected from conservation projects. Now this is massive. The databases out there contain tons of genetic and protein sequence from nearly every scientific study, because it's kind of common practice to deposit your sequences after you use them in one of these databases, which means anybody can then access them and analyse them further. These databases are freely available to anybody at any point. You don't even have to be a scientist, although it would help so you know how to use it. But anyway, you can download it anywhere. So these databases are massive and they require some effort to keep updating and to keep all the data so freely available and accessible. The next way is analysing the genetic sequence to reveal information such as evolutionary history and what taxonomic family the organism belongs to. Now we will cover taxonomy in more detail in the practical lecture, so do not panic. Those of you who have already done the evolutionary bioinformatics module will already know lots about taxonomy, but it's something we will cover in the practical because it is quite important, so don't panic there. So why is any of that important for conservation biology? And essentially it's because we need to decide who we save. There is not an easy question and there is no right answer. There are many different approaches to this question and there is much debate in the scientific community. Conservation projects cost a lot of money and there is only so much funding available. Conservation approaches are very complex. The reason I haven't covered them in this presentation is because they are an entire presentation in on themselves. So one of the most classic ways to view this is species-based approach. Now, to preserve as many species as possible is essentially what the species approach is going for. So preserve threatened species, ecologically important species, species with economic value, species with cultural value. And I found a really great quote, and I don't know who it's attributed to, so if you do know, do let me know. And it is save the last of the least and the best of the rest. That pretty much sums up the species-based approach to conservation. But do you agree with this approach? So do feel free to research other kinds of conservation approaches and make up your mind and debate with other classmates or with friends whether you think this is the right approach that we're taking. So back to bioinformatics. Bioinformatics can help to decide what a species is. It can do this through the construction of phylogenetic trees. This has also been named phylogenetic diversity. The construction of phylogenetic trees is often done in conjunction with ecological studies, such as population geography to assess gene flow. So gene flow is essentially, can the two populations interbreed? This has important conservational consequences. If there is some debate as to whether or not two populations are members of the same species or not, we can analyse their DNA and find out. If they are found to be two different species, they will likely get a separate conservation status, which may impact future conservation efforts, so this is quite important. So we're going to start with a few case studies. I think it's a great way to see it in action. So there was a study carried out by F.A. et al. In 2009. Et al. Just means and colleagues. So originally it was thought that the sandwich tern comprised of one species and two subspecies. Now, if subspecies is something you've never come across before, don't panic. You can always Google it, but all it essentially means is they're not different enough to be a different species, but they can be classed as a subspecies. They're different enough that we can differ them taxonomically slightly, but they are still the same species. Now obviously if they're the same species, they only get one conservation status, so it's kind of important we figure out whether they are or they aren't the same species. So observations revealed minor differences in morphology between the sandwich tern. So we have here the sandwich tern, and this was the parent species they call it, and these are two what they call subspecies. They needed to know, because of the differences in morphology and the differences in breeding habits, whether or not they were the same. An earlier study had looked into relationships between different birds in the tern family, but the sandwich group was left unresolved. So is the current phylogenetic understanding for this group correct? So they collected DNA via blood samples and constructed a phylogenetic tree. They found that the group comprised of two species, the sandwich tern and the Cabot's tern. So what was the outcome? The cabo's tern taxonomic name was changed to reflect the fact that it is a separate species from the sandwich tern, and the authors recommended a revision in the IUCN listing and a conservation assessment on the newfound species. So that's important. We've just gone from one species to two, and they've recommended two different conservation statuses for the both. Another example is the Ethiopian warrior ibex. So this study, they wanted to know whether the similar looking ibex that lived in the same area with them, the Nubian ibex in Ethiopia, whether it was the same species. However, the warrior ibex inhabits only a very small range in the Simien Mountains. It was already classed as critically endangered because of illegal hunting, habitat loss, climate change, disease, competition with livestock. Now, there is only one population left, which that's obviously problems in terms of genetic diversity. Although it was already classed as a distinct species, the previous classification was done primarily on morphology. And I've already mentioned there was some opinion as to whether the warrior ibex was actually a subspecies or not. So they needed to take a look at that as far as conservation efforts go, because that's quite a dire situation to be in with one population left. So is the Ethiopian warrior ibex a single species, or is it a subspecies of the Nubian ibex? Now, the method was to collect DNA from fecal pellets and construct a phylogenetic tree. They also used population genetics and ecological data in the study. The results were the warrior ibex is confirmed as a single species with very low genetic variation and one population of about 450 animals. Going back to our population genetics study slide, why is having a low genetic variation in a single population likely to be a problem for the future of warrior ibex? I will leave that one up to you to think about, but obviously think of small population, small genetic diversity, genetic bottlenecks, and that impact on its future as a species. So what was the outcome? The authors called for urgent conservation actions such as reintroduction into their historic range. Now another one, and this one is slightly cool but creepy. This is a caterpillar fungus, and it is one of the most valuable medicinal fungi on the planet. It's endemic to the Tibetan Plateau. It is used in traditional Chinese medicine to treat a wide range of conditions including asthma and lung inflammation. Trade was legalised in 2001 in Nepal, but since then the local market price has risen 2,300%, but harvests have since declined. The caterpillar fungus is harvested after it is infected, killed, and mummified the larvae of moths, and then produced a fruiting body, which is used as part of the medicine. And that fruiting body is this bit up here. So you've got your mummified caterpillar and the fruiting body up here. It's now classed as an endangered species in China, and this is thought to be due to over-harvesting. There was also some query of the taxonomy of this fungi. A previous study using morphological characteristics had identified 5 potentially new species related to the caterpillar fungus. The Tibetan Plateau is a highly mountainous area, and it was thought likely that gene flow had halted between some populations. So the study question was, what is the genetic diversity and evolutionary history of the caterpillar fungus? The method was to collect specimens and extract DNA, and conduct population genetic studies in a phylogenetic tree. Although these fungi are spread over a large area geographically, the results indicated that it is in fact just one species. Genetic population studies reveal southern isolates had more genetic diversity than northern ones. So the outcomes were the authors recommended the southern, more genetically diverse populations be recognised as important genetic reservoir. Due to the economical and medicinal properties of the species, they recommended that the species be considered for conservation efforts. And that's it, you've made it. Well done guys, we've got through the wordy bit. Do feel free to take a look around the extra resources. I've put all kinds of stuff in there that I used to help build this presentation that I think would be really useful for you to take a look at. I highly recommend going to the California Academy of Natural Sciences page. They give me permission to link to them. I really enjoyed their biodiversity videos. I thought they were fantastic. They complement this presentation wonderfully, and they go into a bit more detail in other things such as ocean acidification. They have 35 short videos. They're all probably less than 10 minutes long, and you can pick and choose the one you want to watch. They are available on the page themselves from the California Academy of Natural Science. They are also available on YouTube, so do check those guys out. And once you're done, head over to the practical exercise and we can get to building our phylogenetic tree and looking between the relationships between some of the big cats. We will then be looking at how bioinformatics has helped shape conservation efforts for 2 of the endangered clouded leopard species, like this guy down here. See you there guys!